Hi, my name is Sarita, lecturer in Botany, Government Degree College for Women, Nalgonda. Today in this video I am going to talk about Pinus stem anatomy. This Pinus belongs to division Gymnosperms, class Coniferapsida, order Coniferales, family Pinaceae, genus Pinus. Here I am going to talk about primary and secondary structure of Pinus stem. The young stem of Pinus resembles the dicard stem anatomy. Coming to the surface of the Pinus stem, it shows uneven surface. Pinus stem surface is uneven surface. Transverse section of the Pinus stem shows mainly three parts. They are epidermis, cortex, and vascular cylinder. Coming to the epidermis, this is the epidermis. It is outermost layer, 1 cell thick without any intercellular spaces. This epidermis is covered by thick cuticle layer on the outer side. Cuticle layer present at the outside of the epidermis. This epidermis is the protective layer. It protects the innermost parts. This is about epidermis. Next, cortex. Beneath the epidermis, cortex is present. It is differentiated into hypodermis and general cortex and endodermis. Beneath the epidermis, cortex is present. This cortex is differentiated into hypodermis, general cortex and endodermis. Coming to the hypodermis, beneath the epidermis, hypodermis is present. This is the hypodermis. It is made up of sclerenchymatous tissue. Below the hypodermis, general cortex is present. This is the general cortex. This is the general cortex. Below the hypodermis, general cortex is present. It is made up of parenchymatous tissue. This cortical zone is chlorenchymatous and also possesses numerous resin-kinase. These are the resin-kinase. This general cortex possesses numerous resin canals. These resin canals are schizogenous type of resin canals. Here, schizogenous type of resin canals are present in this Pinus stem. Schizogenous type of resin canals means schizogenous cavities forms the schizogenous type of resin canals. Schizogenous cavities are formed by the enlargement of intercellular spaces. These cavities are lined with epithelial cells. Schizogenous cavities are formed by the enlargement of intercellular spaces. These cavities are lined with epithelial cells. These are the epithelial cells. These cavities are lined with epithelial cells. These epithelial cells secrete the turpentine into the resin canals. This is about general cortex. This is about general cortex. Next, beneath the general cortex, endodermis is present. Last layer of the cortex is known as a endodermis. It is a limiting layer of the cortex. Because of the absence of Casparian strips, the endodermis becomes obscure. This is about cortex. Next, vascular cylinder. Third part is vascular cylinder. Below the cortex, vascular cylinder is present. This is the vascular cylinder. Below the cortex is the vascular cylinder. This vascular cylinder enclosed with pericycle. This vascular cylinder enclosed with pericycle. This vascular cylinder consists of 5 to 8 vascular bundles in a ring-like manner. They lie close to one another. Thin medullary rays are present in between the vascular bundles. These vascular bundles are conjoint, collateral, endarc and open type of vascular bundles are present in this Pinus stem. Vascular bundles are conjoint, collateral and open, and endark type of vascular bundles are present here. Each bundle has phloem outside and xylem inside. Collateral vascular bundles means when xylem and phloem are present side by side on the same radius, the vascular bundles are known as a collateral vascular bundles. In this collateral vascular bundles, the xylem is present towards the inside and phloem present towards the outside. Open type means here we can see open collateral vascular bundles, closed collateral vascular bundles. What is the difference between open and closed? In this, coming to the open collateral vascular bundles, in between the phloem and xylem there is a cambium is present. In between the phloem and xylem, cambium is present. This type of vascular bundles are known as a open collateral vascular bundles. Closed means in between the phloem and xylem there is no cambium is present. Here cambium is absent. The type of vascular bundles are known as a closed collateral vascular bundles. 2 to 3. Here open collateral vascular bundle. This is the open collateral vascular bundle. Here xylem present towards the inner side and phloem present towards the outside. 2 to 3 cell thick cambium is present in between the phloem and xylem. Here 2 to 3 celled thick cambium is present in between the phloem and xylem in this open collateral vascular bundle. Protoxylem. Protoxylem means protoxylem is the first formed part of the xylem is known as a protoxylem. Metaxylem forms later. Protoxylem tracheids are with spiral thickenings on their radial walls. Metaxylem tracheids radial walls possess border pits. Xylem consists of tracheids only. Phloem consists of sieve tubes, phloem parenchyma, and component cells are absent. Albumin cells are substitute the component cells. Albumin cells substitute the function of component cells. The central part of the stem is with a small amount of medulla. Which also contains resin canals. The central part of the stem is with a small amount of pith, small amount of medulla, which is also contains resin canals. This is about primary structure of finus stem. Next, secondary growth. Secondary growth of finus stem resembles the secondary growth of dicot stem. Parenchymatous cells which are present in between the vascular bundles. Parenchymatous cells which are present in between the vascular bundles of the vascular cylinder transform into meristematic tissue and forms the interfascicular cambium. This is called interfascicular cambium. This interfascicular cambium combines with the fascicular cambium. This is called fascicular cambium. Fascicular cambium present. This fascicular cambium present in between the phloem and xylem. Interfascicular cambium present in between the two vascular bundles. This is the difference between fascicular and interfascicular cambium. The parenchymatous cells which are present in between the vascular bundles transform into meristematic tissue. This meristematic tissue forms interfascicular cambium. This interfascicular cambium combines with the fascicular cambium and forms cambial rings. These are the vascular bundles. This is the phloem part, this is the xylem part. Here this is the cambium which is present in between the phloem and xylem is known as a fascicular cambium. Next, interfascicular cambium is the cambium, some of the cells which are present in between the two vascular bundles transform into meristematic tissue and forms interfascicular cambium. This is called interfascicular cambium. This interfascicular cambium combines with the fascicular cambium and forms cambial ring. This cambial ring takes further divisions and forms secondary phloem outside and secondary xylem inside. Secondary xylem is pycnoxylic type of xylem is present. Pycnoxylic means xylem tracheids are small and thick walled, along with the xylem parenchyma. Xylem rays are very few and forms narrow xylem rays in the pycnoxylic xylem. Medullary rays join the secondary tissues. Annual rings are very distinct in the secondary xylem. Annual rings are composed of spring wood and autumn wood. Spring season is more suitable for the growth of the Pinus plants. That is where secondary xylem cells forming in this season are large with thin walls and supply water and minerals quickly. This xylem is called spring wood or early wood. This is the spring wood. In winter season, that is unfavorable conditions, very less wood forms and the xylem cells or tracheids are narrow and thick walled. This wood is called autumn wood or late wood. This is autumn wood or late wood. Each year, the cambium forms a bond of secondary xylem and secondary phloem. In cross section, it appears as a series of concentric rings called annual rings. In cross section, it appears, this spring wood and autumn wood appears as a series of concentric rings called annual rings. Counting of the number of these rings gives an indication of the age of the tree. Annual rings indicates the age of the tree. If the tree shows 12 annual rings, that means the age of the tree is 12 years. Xylem cells of the secondary xylem are arranged radially and border pits are present on their radial walls. These border pits can be in one row or in two rows. Biseriate pits are arranged in an opposite manner and are called Obtainian pits. Bars of Sanyo are present between the pits and these are called crassulae. Secondary phloem consists of sieve cell and sieve tubes, phloem parenchyma and phloem fibers. Companion cells are absent. Albumin cells substitute the function of companion cells. In Pinus, the secondary medullary rays are structurally more complex. Medullary rays in secondary xylem are called xylem rays and the medullary rays in secondary phloem are called phloem rays. These phloem rays and xylem rays are formed from cambium cells. When the secondary growth takes place in vascular cylinder, outside the cortex, cork cambium is formed with continuous division. This cork cambium this is the cork cambium. This cork cambium forms the secondary cortex inside and cork outside. When the secondary growth takes place in vascular cylinder, cork cambium forms outside the cortex with continuous division. The cork cambium forms the secondary cortex inner side and the cork outside. Secondary growth in the cortex results in this crushing of the epidermis to form cork and bark outside. This is about Pinus primary and secondary structure. This is about Pinus primary and secondary structure of Pinus stem. Thank you.